We will now hear from a few other projects in the field tackling the problems with pests and Xylella. So we have Maria Saponari from the IPSP CNR, she's senior researcher. We have Giuseppe Lima, who's full professor at the University of Molise, and Stéphane Compin will be also presenting the BIOVEXO project, or representing rather the BIOVEXO project, not presenting, not re-presenting either. No. All right, so first we'll be hearing then from two projects from Maria Saponari, the BeXyl project and the OMIBREED project. So please welcome Maria.
No? Okay, now, okay. First of all, I bring the apologies from Blanca Landa. She's the coordinator of the European project BeXyl. She could not travel and attend the meeting, but she sent a few slides just to present here what has been done in the BeXyl project, since this European project is also almost at the end. Next year it will be the final report of the project. I asked the technician to launch. She sent a video that summarizes a little bit the project, and then I will show just few slides regarding the complementary actions of this project with BIOVEXO. Presentation, I can add only a few more words regarding the topic of this project. This was the first project funded under the Horizon Europe program. Okay, sorry. Okay, sorry. The consortium is quite large. We are 28 partners from lots of countries, as you see here, not only European countries, but we have also third countries. Their participation, although is not funded by the European Commission, was considered highly important to exchange information and knowledge from these countries where the bacterium is already well established, or as is infecting crops from which we need information in Europe. Regarding I go fast as we said, the project include different objectives, and so it's not only the curative method differently from BIOVEXO that was targeted on curative methods and preventive methods. The project also include the strengthening of measure to protect the European Union from the introduction of Xylella and for the further spread. So there is relevant activities on improving surveillance and modeling for prediction of the spread, and so to strengthen all the measures for preventing severe impact of the pathogen. Sorry. Okay, and the final objective of the project is to have an IPM strategies, because it's quite clear that we cannot tackle Xylella disease with only one action or one solution. So the objective of the project, as it was also in BIOVEXO, is to put together an integrated strategies to counteract the impact of the bacterium, which include the resistance of the host plants, but also action and solution against the bacterium and the vectors. One peculiar point of the project is that during the project it has been set up a network of sentinel plots. So in all these countries, through the project, there will planted a set of defined species which have the role of sentinel plants. So these include outbreak area or Xylella-free area. In the outbreak areas, the objective of this plot is to see which is the species that on which we can early detect the infection, and so a plant species that should be regarded as a sentinel plants. Whereas in the Xylella-free area, the scope and the aim of this plot is to eventually intercept introduction and establishment of the bacterium, which has not been yet detected. I will not go in depth in the IPM strategies, because Professor Gianni Giglioli in the next session will speak about it. But I will mention some aspects that are complementary to those presented in BIOVEXO. In fact, when the project was prepared, it was paid attention to be complementary and to do not overlap or redo work that was already done in other European actions. So, for example, for the vectors, the project concentrated on the use of some endophytic fungi, and this is some of the illustration. Sorry for that. I will not go in depth in the data because of course there is no time, but and these were both tested in the field with the involvement of the group of Enza Don Giovanni, both for young, for the juvenile, and for the adults. Another aspect that is complementary to what we already heard this morning is the study of the virome associated with the insect. So to study if some of the insect that has manifested some alterations could be related to the presence of some viruses, which could be used for our purposes to reduce the fitness of the insect. And this work is carried out, is under the leadership of Professor Domenico Bosco in Torino. As well as he is also leading the work on searching for parasitoids, which unfortunately seems not to be a good route for the control of these insects, as a low number of parasitized eggs or adults were collected during the surveys carried out during the project. And the third action that is included for the vector control is the agronomic management, and these activities are ongoing in Spain and in Italy, in grapevine in Spain and in olive in Italy, as also carried out by the group of Enza Don Giovanni. And regarding the control of the pathogen, the innovative tools that I want to underline in BeXyl is the development of a synthetic community of microorganisms. Knowing that the Xylella has a complex interaction with the microbial community in the plants, and the difficulties to control this pathogen in the plants, we know that it would be a dream to identify only one antagonist. So the idea behind this task is to find a consortia of microorganisms that can protect, mitigate the impact of the disease in the plants. So I think that next month we will start the field trials with some of the microorganisms that have been isolated either from the plants and from the vectors to be used, to be tested in the field here. And the same there is also in our project the testing of antimicrobial plant extract, whereas one of the innovative approach is the use of peptides. These were already tested in previous project, limitedly to in vitro test, but in BeXyl we are now in the field stage. And yes, I will not go, as I said, in the integration of these methods and how they are applied in the field, because Professor Giglioli will explain later. So thank you, and these are the reference of the project. I will continue now with the project that actually I coordinate. Is a national research project, and it deals with the resistance on Xylella fastidiosa in olives. It's the project OMIBREED. This project has been funded in 2023 along with another two projects which target the study of the resistance against Xylella fastidiosa. We know that what is the resistance in two words. We know that once a plant gets infected by the vectors, there are some plants which react to the infection, and the bacterium, even if start multiplication in the plants, the colonization is quite limited and restricted in the canopies, whereas the susceptible plants are intended those that succumb to the infection. So as you can see in that drone, the bacterium is able to digest the pit membranes of the xylem vessel, and so to easily colonize the plants and fully invade the plants. At the moment, the mechanisms that occurs in the resistant plant, as well as which are the host determinant of this resistance, are not yet known. There is more knowledge in grapevines, where the studies have a long history in US, but very little is known, for example, in olives or yet in almond. So the starting point for this research was the evidence since the beginning of the epidemic in Salento that there were some plants that survived the infection. They are not immune. We are not speaking about immunity, but they suffered much less than the susceptible one, and they continue to produce. So in these past 10 years, lots of research have been carried out, investigated different aspects of the plants to understand if it's an anatomical difference, physiological or genetic, have a genetic basis, or the metabolic profile, or if these plants harbor a different microbial community. So far, unfortunately, the evidence that we collect is that the genetic traits associated with the resistance in olive are very limited in the germplasm. So there is only a small cluster of genotypes which harbor these traits. And exploring the natural genetic resource in the epidemic area, I mean the wild olives in the infected area, we also confirmed that if there were some plants showing resistance, not suffering from the infection, most likely they all derived, if not all, most of them derived from Lecino. So they were seeds of Lecino cultivars. What's the project? Which are the main objective of the project? The first task that we have just completed during these second years is the collection of a wide range of olive genotypes to collect as much as possible the genetic variability that exists in olives. And the main attention was paid to the ancient olive trees so that we can capture as much as possible the variability that occur in the Mediterranean basin, where the origin of this plant species is. Sorry. And what we will do with all the data, the genetic data that we accumulated with all these genotypes, the group, the genetician, are characterizing these plants in order to identify unique genotypes. In olives, it's quite often to find synonymous or plants that are identical but named with different names. And so the final objective is to have a map of the unique genotypes and then combine these data with the phenotyping data. For phenotyping data, I mean how these plants react to Xylella or other stresses. Of course, we cannot test 1,000 different variety for Xylella because the efforts that are needed, either if we do the screening in the field or even in the greenhouse, is huge efforts and take long time. But on a small subset of cultivars, we are producing this data so that they can be matched with the genetic data to find which are the conserved sequences across the different cluster of cultivars. We are not working only on Xylella stress, but this abiotic stress, but also on abiotic stress, like determine which is the cold resistance of the cultivar. This is a quite easy phenotypic data to acquire because it's quite fast, as well as we are doing also a work to determine the response to drought stress, because it is another important factor nowadays. The other points important from the project to identify which are the genetic determinants associated to the resistance is to use segregating population. So we have population crosses derived from the resistant Lecino and the susceptible Oliarola, and we hope that in the near future, we can get some information by characterizing the segregating characters in the population. All these plants are genetically characterized, so once we know how phenotypically these plants react, we can again associate the genetics to the phenotypes, and so in the hope to associate and to identify the genetic region of the plants that are responsible for the resistance. It's also important nowadays to have an interaction between the different approaches that the new era of genomics offers. So one of the important task of the project is to combine different approaches on the same set of plants, and this is very important. Then all these tasks that I presented are finalized to understand the mechanism of resistance. Then the project has also the objective to produce a new resistant germoplasm. This, at the moment, can be done through conventional breeding. As you can see here, several crosses have been done during the past years, including at least one potential parental with resistant characters, so that we can hope that in the progenies we have new plants with these which harbor the resistance as the parental. The bottleneck is that we need to carry out this large breeding program. We need field plots, and we have to thank the Onlus Save the Olives, because they are collecting funds to and make available plots for the research. And these are also some of the plots that have been already realized to start to test this new germoplasm. This has been done in collaboration with a regional agency. And just in few words, this slide to summarize that this is a process to produce and to generate a new cultivar for olive. We need at least 12, 15 years. So there is a need for long-term research. This is the example in olive to register a new cultivar resistant to verticillium. In the case of Xylella is even more complicated, because to infect and to assess if a plant is susceptible or resistant to Xylella, it takes several years compared to what is done with verticillium. And so with this project that lasts only 3 and 4 years, we could not make all the program, but we are capitalizing all the previous investment in this direction on the resistant studies, so that we can advance the activity. We know that we will not reach at the end a new variety, the activity will be in progress, but and we have to thank for this program all the collaboration between private association and the research institute, because without having a large facility and plots available, it's quite difficult to carry out this activity, as well as several organizations in the Mediterranean countries which helped to collect the material. And I conclude with the information of the dissemination activity and the communication that you can find on our website. Thank you.
Thank you so much, Maria. Please, you can take a seat, and now we'll be hearing from another project, Integroliv, with Professor.
Here. Good afternoon. Integroliv is a national project supported by the Italian Ministry of Agriculture. Is an interdisciplinary and multifaceted project that aims to develop an integrated approach to control as a main goal to control Xylella fastidiosa on olive plants, but also to act in the olive agro ecosystem to control also other olive pathogens. Obviously, for an also economical point of view, to reduce costs of plant disease control in the agro ecosystems. In the projects, there are 4 research units. The university plant pathology group of my university, University of Molise, 3 National Council Research Institute, and the University of Bari, and also University of Naples. We have 7 work packages, and 4 of these, from work package 2 to 6, are research work packages. And the first is the management of the project, and the WP7 is the activity that now we are made, the communication and dissemination of results. The work package 2 aims to evaluate, to screen a large numbers of products, in general sustainable products, biological control agents, for example, natural compounds, commercial formulates of these products, but also experimental products. This activity, now we are at the 2nd year about of activity, and then the activity of this work package was concluded, and we have selected several products of various types, and these products are now evaluated in screenhouse, under quarantine screenhouse conditions, in the field, on different varieties of olives susceptible to Xylella fastidiosa, such as Oyarola and others, both on young plants, but also on old olive plants. In particular, in the infected area, we are conducting 3 experimental fields, 1 with young plants and 2 in different locations, and 2 on old olive plants. In the work package 3, the work package 3 regards the evaluation in planta, on potted olive plants, of products that we selected in the work package 2, in vitro and laboratory screening. And this activity is still in progress under greenhouse conditions. In contemporary in the VP4, we are studying the mechanism of action of the most promising products in regards to the activity on the pathogen, but also in the interaction with plants and in the multitrophic interaction plants, pathogen, and products. And also this activity is in progress, as well as the field trials that are conducted from 1 year in the field. As you know, the cycle, as also Maria said, of Xylella fastidiosa disease in plant is very long. And then we, the first year, have applied several treatments, about 6-7 treatments of single products or also combined products in protocols, specific protocols. And now we are evaluating evaluating symptoms, but also the colonization of the bacteria inside the plants. And this activity is carried out on younger plants in a field where we are evaluating experimental products. But on all the mature plants, we are evaluating commercial formulates of different products that we selected in the previous work packages. These activities are developing in cooperation with two assaying center, the assaying center of Basile Caravia, of this research center, and also the Biofarm of Ercolano, Napoli. Results, the experiment are in progress. We hope next year to acquire good results. The work package 6 involve a committee of experts of our team to set up, to choose the better protocols that we set up in the project in order to propose these protocols for practical application in the field, we hope in the close future. The work package 7 regards the dissemination and communication of results. For further information, we also have a website that you can visit to acquire more information about. An important aspect is that our protocols can be also very useful not only for susceptible varieties of olive plants, but also for tolerance, because with the new system of growing with plants more close in the field each other, we expect to have more impact of other diseases, fungal disease, but also other bacterial disease. And moreover, these new varieties, as Lecchino, for example, and others, are tolerant but not immune to the Xylella. As you can see also here, some symptoms of recent symptoms on Lecchino that we are observing in the field. And we have to don't forget that olive tree is not for olive and oil production, but is also connected to history, culture, tradition, landscape of a territory as the Apulia region. We have a century and also plants that have thousands of years. The effective control of Xylella on olive plants is very, we are aware that is very, very difficult and an ambitious challenge. However, as Albert Einstein said, it is in the crisis that the best of each person, in our case researcher, emerges, because without the crisis, all wines are just gentle breezers. The Integroliv project will most likely not solve the problem Xylella, but it will help to establish a sustainable coexistence in the infected area. Our project is addressed particular to infected area, between the pathogen and the olive trees. These are all the researcher colleagues that are participating on the project team, and also we give many thanks to our younger staff, our younger co-workers that are cooperating on all activity with very high enthusiasm. Thank you for your attention.
Thank you so much. Please take a seat. So you've now heard from effectively 4 projects, BIOVEXO, OMIBREED, BeXyl, and Integroliv. So different projects, different approaches as well on how to tackle Xylella. And I would like then now to get a bit of feedback from you. If we can just show the audience how to connect. So what I want you to do is whip out your phones, get your phones out. And you can connect here to, very quickly, or very soon, there will be a QR code, or you can go to a website called menti.com. Can we put it up? You have it? Yeah, okay. It'll be coming in a second. Here we are. Okay, so you have a QR code here, or if you're not a big fan of QR codes that then kind of track you to the bone, you know, they will track you wherever you go from hereafter. A little bit like Xylella in a way, you know, tracking you down. You can also go to menti.com and use the code 4800-1246. Is everyone good? I mean, you may not all be connected yet, but you're almost there. No problems? You're all in? Ah, I'm in the way. Ah, yeah, look at that. Plenty. Plenty of feedback. Great stuff. Okay, so as you connect, I want to invite you to a first reflection, right? I want you to imagine that you are, let's say, the European Commission, or at least an organization with a lot of money. Okay, are we ready? Can I get a yay if we're ready? Okay, is anyone not ready with a nay? No. Okay, we're good. All right, let's do it. So, here we are. Next slide. No, it's not. You don't have it here? This should work for me. No? No? It doesn't show? Okay, yeah, go for it. You do it. Yeah, I disconnected from the internet, that's why. Okay, this is completely pointless. Okay, so I want you to imagine you're the European Commission, and you have 100 million euros in your pocket, okay? And you have to decide where you want to put your research budget. Combat Xylella. Now, usually when I would ask a question like this, people say, oh, we need a bit of everything, you know? That's why I'm asking the question in this sense. Here, you can actually fund everything if you want to, but you only have 100 million euros. So what you need to do is to split your 100 into these 4 different options. You have genetic resistance and harnessing plant microbiome, you have integrated pest management, advanced detection methods, or biological controls. Now, don't think about it as how much will it cost. Think about it more in terms of what is a priority, okay? Because maybe you wouldn't put the same amount of money to different things because they just cost more or whatever, or the research is more complicated. Forget about that. Forget about the money in that sense. Take it as a prioritization exercise, okay? And I would like people to vote, like maybe go up to around 30 odd people. That would be good. We'd be around half, just to get a sense of where the audience sees the importance of these different aspects, because I think you've all touched upon some of them in your various projects. You've taken a direction, let's say, and I think it will be interesting to hear also from the audience on this. It looks like, for the moment, genetic resistance, integrated pest management are the top players here, and biological control and advanced detection methods a little bit less. Maybe a first reaction from Giuseppe, if you can. I think you took an approach that's more on integrated pest management, a little bit more holistic. You then have to sort of agree with coexistence. Could you maybe just comment then on this? Like, how much do you think integrated pest management is where you would also put your money, and how much do you think other aspects should be considered as well? And please click on the button. It's okay.
In my opinion, all strategies should be combined in an optimal way, because we, against a so serious problem, we have to use synergies of these means and strategies. For example, genetics, the genetic improvement is important, but also is important to adopt all practices to keep plants in a good condition of resistance, environmental resistance, other than gene resistance. The genetic improvements, we have, don't forget that plants is made by genes, but also the pathogen have genes. And then in the future, perhaps the situation will evolve. Mm-hmm. And then we can have biotypes of the pathogen that overcome this resistance, mm-hmm. that the genetics will develop. And then we have really other strategies to reduce risks that the plants can be again susceptible to the pathogen and are touched seriously, as for autochthonous varieties actually.
Yeah. And well, we can't see it now, but basically genetic resistance and integrated pest management are clearly the top two. Stefan, I mean, obviously you're working more on the biologicals aspect here, but you need to somehow connect to the genetic resistance as well, right? I mean, varieties will develop. How much do you think these two issues can combine?
We need to work on all these aspects. We cannot work individually only on one. All these research topics are necessary to develop to fight against Xylella. There was some talk saying not only one solution can reduce the disease, and we need several approaches. We need diagnostic tools to first detect the Xylella. We need some biocontrol agent, not only one, several one. We want to have more tolerant variety, but also to study the effect of our biocontrol agent on to have more tolerant of the tolerant variety. Also the plant microbiome, I'm working on that. And of course, IPM strategy, because you have seen also in BIOVEXO with curative approach and IPM strategy, we have different results on the Xylella population. In the case of curative, there is no effect on the Xylella population, and on IPM, we have a reduction. So all these aspects are necessary to study, and honestly, it will be better to have 25, 25, 25, and 25% of each, because we are not expert on all these kind of things, but all together we can maybe change the situation in the future.
Yeah, and I mean, actually, it's not even quite a percentage. I mean, everyone's sort of placing it. It's a very tricky one, but I think it just gives a picture of the breadth. And if we get, let's say, most people thinking the priority is, let's say, genetic resistance. So to come back to Maria's projects as well, it's also probably the one that will take the most time. And that in that time, many other things can happen.
Yeah.
So how do you manage that in terms of the research to project yourself in what will only come in minimum, let's say 15 years, but by then maybe a lot of these other tools have developed and it's another kind of genetics that you need again?
One aspect I want to remark before answering your question is that, as I showed, the resistant plants are not plants immune to the infection, so they harbor the bacterium, and this means that if these plants are under other stresses, not well managed, or as Professor Lima was mentioning, being under multiple stresses, they can also express more severe symptoms than usual they do. So it is important, the IPM concept is also in this direction, that we need a resistant plant, but being a specific type of resistance, which is not total resistance against the bacterium, we need to combine with other strategies to maintain the plants in the best condition so that we have a resilient plant. It's not enough to have a resistant plant, but we have to grow the plants under the good condition. If the bacterium, if we are in an epidemic area where the bacterium continue to circulate and we have a pressure of inoculum. Regarding your question, it's important to say that maybe even at EU level, the concept on the genetic biotechnology and genetic transformation is a concept that start to be more accepted, and so we have some project which started to explore also in olive the new technologies for genetic improvement, which we, at the moment, we cannot forecast to develop new plants in short time, but we believe that once the technology are optimized in olive, we can shorten that period that for conventional breeding now is more than a decade.
And maybe, I mean, we touched upon this towards the end of the previous session on the interaction, right, between research and business. You want to add something? Please, quickly.
If I can add, obviously the biological control, in my opinion, is included in the integrated pest management. And for the last point, advanced detection methods obviously are very important, but we have to distinguish infected area from non-infected area. For non-infected area, advanced detection methods are very, very important in the view that preventive methods are better than curative methods when possible.
Yep. Okay. And thanks to everyone who participated. We will come back to these things, so don't worry. I know it's fun. But one thing I wanted to touch upon, because at the end of the previous session there was a bit of conversation around the interaction between research, business, uptake of research results, and how much research results can be shared. I would also like maybe a quick reflection on how the different projects have managed that interaction or are managing that interaction. How much are you interested in actually getting products in the market in the end, and how you think you can achieve that. I mean, Stefan, obviously that's a core part of the project because it's a kind of requirement in a way, or ideally. So I think, can you just say a word maybe about how you've managed that or how you intend to manage that?
Yeah, at the beginning our goal was to help the farmer and to reduce the Xylella symptoms. So as a scientist, in my opinion, we give a service for the society. And of course, we made a lot of publication, but not only publication, we need to have end product to help the farmer with the situation. So it's both science and go to the final product from the basic knowledge to the end and to be on the market, in my opinion. But I'm working in an RTO, so I do all these kind of things.
And Maria, in your case, can you maybe comment a bit on the interaction, if any, with the business side?
Yes, in our case it's not exactly in the business side, but as I mentioned, the public and private partnership for this long-term project is quite important, because developing new variety does not mean that we have only to take care of the resistant characters of these plants, but also the agronomic feature of these plants, if these are then accepted by the consumer, by the producer. So the interaction with the producer to do a large-scale validation, and so to use their farm to do this activity is critical, it is essential, and so also for this in this line there is there are agreements to develop together these long-term projects.
Okay, thank you. Giuseppe, in your case, is there any interaction?
As before I evidenced, an important aspect is the economic aspects. And then all of these topics are important, but we have to consider that the costs of the farms that will be increased to control this unexpected pathogen in this last year. And then all new measures that I suggest for practical application, we have to need to perform an economical evaluation. Then in our project is different from others because as the goals also all pathogens of olives together with the Xylella fastidiosa. And then when our products were evaluated also for side effects also with fungal and other bacterial pathogens. And then when farmers apply a treatment, this treatment is not only specific for Xylella, but also for other pathogens. And then cost of control decrease. The economical aspects is very important.
Okay, thank you. Any questions, comments? No? All clear? All good? All right, so in the sake of time, I would like to then thank the three of you for having shared your views. We can give them a warm hand.